Hello and welcome to all my dear students. So, today we are going to start with the next lecture of the chapter Genetics Part 1, Principles of Inheritance and Variation. So far beta, we have discussed about the work of Mendel, including monohybrid and dihybrid cross. I hope that portion of the lecture you have gone through and you have understood well also. Then we also started with two-gene interaction, where I was telling you that how scientist Bateson, also known as father of modern genetics, started or expressed that even two genes located either on same or different chromosome can control one character. In that you have seen two examples of complementary gene interaction and duplicate gene interaction. Now, apart from this, in my last lecture, we have already discussed about the concept of epistasis and how epistasis is different from dominance. I hope you have understood it that epistasis is basically intergenic or non-allelic interaction, whereas dominance is intragenic or interallelic interaction. Now, epistasis can be of two types, dominant and recessive epistasis. So today basically we are going to study about what is dominant epistasis and what is recessive epistasis with the help of example. Then we will try to understand one more exception of Mendel, that is polygenic inheritance. Then we will start with chromosomal theory of inheritance, and in the last we will get some introduction about the concept of linkage proposed by T.H. Morgan. Correct? So this much are the things which we have to do today. Not much to discuss, but yes, whatever we are going to discuss is very, very important. So let's start with dominant epistasis. The example which we are going to study under dominant epistasis is the fruit color of Cucurbita pepo. Cucurbita pepo is a member of family Cucurbitaceae. Now, if you check the fruit color of Cucurbita pepo, then there are three distinct fruit colors: white, yellow, and green. So, how many colors are there for Cucurbita pepo? There are three distinct colors: white, yellow, and green. Now, this fruit color of Cucurbita pepo is one character, but under the control of two genes represented as W and Y. Correct? Now, they show dominant epistasis. That means one gene in its dominant state will be epistatic and will suppress the effect of another gene, be it present in dominant state or in recessive state. So over here beta, this W gene in its dominant state suppresses the phenotype of Y gene. Correct? Yes or no? Now beta, W that means is the epistatic gene, whereas Y is the hypostatic gene. Clear? Now, there are different types of genotypes possible for W and Y gene. Now, according to Mendel's dihybrid cross, the expected ratio would have been 9:3:3:1, where this 9 stands for where both the gene are in dominant state, maybe homozygous dominant or heterozygous dominant. Next 3 indicates that one of the gene is in its dominant state, then another is in its recessive state. Next 3 indicates the same, and last means both the genes are present in their recessive state. So this is the expectation. Correct? Now what here will happen beta, wherever you see W as dominant, maybe homozygous or heterozygous. Now in that case, the fruit color of Cucurbita pepo will become white. So let me tell you one thing, that if W is dominant, maybe homozygous or heterozygous, then the fruit color will be white. If W is present in its recessive state, whereas Y gene is present in its dominant state, then you know there is nobody to suppress the Y effect. So Y gene in its dominant state will produce its proteins and coloration will become yellow in that case. What will be the color in that case? It will be yellow. Now suppose if both the genes are present in their recessive state, then neither white nor yellow. So the color will become green. So now just check it over here. Here you can see W is dominant. Now whatever be the condition of Y, no worries, color of the fruit will be white. Here you see beta, W is in its recessive state, but Y is present in its dominant state. So definitely the color of the fruit will become yellow. Here fruit color will be white, but in last case you see both the genes are present in their recessive state. So the fruit color will become over here green. Now, thus the ratio 9331 for dominant epistasis changes to 12:3:1, where 12 means 12 white, 3 yellow, 1 green. I hope you have understood the ratio well and which number indicates which coloration of the cucurbita pepper. This is about dominant epistasis, where one of the gene in its dominant state is epistatic over another gene. Now, second type of epistasis is basically recessive epistasis. What happens in recessive epistasis, beta? In case of recessive epistasis, the epistatic gene is present in its recessive state. Here you saw that the epistatic gene is in its dominant state, so dominant epistasis. However, if the epistatic gene is in its recessive state, then that type of epistasis is recessive epistasis. We will understand the concept with the help of an example, coat color of mice. Coat means skin color of mice. Correct? Now beta, this coat color of mice is under the control of two genes, C and A. Right? Now, C gene beta in its recessive state is epistatic over A gene. Right? Is epistatic over A gene. That means this A gene is actually responsible for coloration. Right? Now, if A is present in its dominant state, maybe homozygous or heterozygous, then the color of the coat in mice will be a gouchy, which is slightly grayish in color. However, if A is present in its recessive state, then the color will be black. Now, whatever be the arrangement of A gene, if anywhere C is present in its recessive state, then in both the condition it is going to suppress the effect of A gene. Neither organism will come colored or black or a gouchy, rather the mice will become albino, which is colorless. So basically C gene in its recessive state does not allow A gene to give color to the coat color of mice. A gene alone present if C is there in its dominant state, then dominant C is doing nothing. It is letting A gene work. So A in dominant state a gouchy color. A in recessive state along with dominant C, which color? Black color. But if C is in its recessive state, then no coloration, skin color of coat will become completely albino. Understood? So now what is the condition? How is the genotypic ratio seen? Ratio for dihybrid crosses 9:3:1. How do you write it? First, where both the genes are in dominant state. Next, where one of the gene is in its dominant state. And last, where both the genes are in its recessive state. So tell me, over here, what will be the coloration? You see, C is in its dominant state, but not epistatic, because here it shows recessive epistasis, not dominant epistasis. So A is going to function and A is present in a dominant state. So the color of the coat will become a gouchy. In next, C is in its recessive state. So recessive C will become epistatic over A, will not let A function, and the mice will become albino. Next 3. C in its dominant state, not epistatic. A is present in its recessive state. So color will become black. And last again you see, C is in its recessive state, so it is going to suppress the effect of recessive A, and hence the organism will again become albino. So how the ratio gets modified? 9:3:4. 9 organisms will be a gouchy, 3 will be black, and 4 will be albino. Understood that what is the ratio for recessive epistasis and dominant epistasis? If till here things are clear, absolutely great. Now we are moving ahead with the next exception of Mendel, that is polygenic inheritance, where I'll be teaching you the example of human skin color. This concept of polygenic inheritance in human skin color was explained by scientist Davenport. Now, beta, polygene is just the reverse of pleiotropy. Pleiotropy simply means when one gene is controlling more than one character. Polygenic means where many genes are controlling one character, right? So what is polygenic inheritance? Polygenic means many genes controlling one character. Correct? Yes or no? Many, genic, gene. Many gene controlling one character, like for example, human skin color studied by Davenport is controlled by 3 genes. It's controlled by a pair of 3 genes. That is A, B and C. Right? Now beta, like you saw in case of incomplete dominance, polygenic inheritance is also quantitative inheritance, where number of dominant alleles, the quantity of dominant alleles is going to decide the phenotype of an organism. So basically, polygenic inheritance is also a type of quantitative inheritance where number of dominant alleles play major role, not the quality of dominant allele. Clear? Yes or no? Chalo. Now let's try to understand that how human skin color shows polygenic inheritance. First is because it is under the control of 3 genes. 3 genes, they control one character. So obviously it is polygenic. I'll just try to make a slight cross to show you the polygenic inheritance and how number of dominant allele play important role in deciding the human skin color. Okay, now 3 genes means if all the 3 genes are present in their allelic form, suppose it's a diploid organism, then first of all, how to start the cross? To start the cross, first step is selection of pure line parent. Selection of pure line parents. So one parent has to be homozygous dominant for all the alleles and another has to be recessive for all the alleles, for all the genes, right? Suppose this is male parent and this is female parent. Beta, remember, more the number of dominant alleles, more will be the synthesis of melanin pigment and darker will be the skin. So any person who is having all the 6 dominant allele, 2A dominant, 2B dominant, 2C dominant. So here you see 6 dominant alleles. So here there will be maximum synthesis of melanin. There will be maximum synthesis of melanin. As a result, this person will be a Negro, a black person. Correct? Now, this Negro marries a woman who is having all the 6 recessive alleles. 2A recessive, 2B recessive, 2C recessive. That means no dominant alleles. 0 dominant alleles. That means no melanin synthesis. That means, beta, no synthesis of melanin. Hence, the skin color will be absolute white. You can call an English person, like a Britisher or an American, their skin is completely white. They don't have melanin production. Because why? As all their alleles are present in recessive state. Now, if an African and an English woman and man marries, then what is the highest chance for their offspring to have which type of genotype? Obviously, now gametes will be, male is homozygous dominant for all the 3 genes. So gamete only one type, that is this. And female is homozygous recessive for all the 3 genes. So number of gametes produced will be 1, which is small a, small b, small c. Now if these gametes, they fertilize, beta, then all F1 generation offsprings will have heterozygous arrangement for all the 3 genes. That means how many dominant allele? 1, 2, 3. So basically, there will be 3 dominant allele. So what will be the coloration? Coloration will be neither too dark nor too white. It will be an intermediate. 6 dominant means maximum melanin. 0 dominant means no melanin. 3 dominant is an intermediate condition. So intermediate amount of melanin will be synthesized and such color is known as mulatto. Like you must have heard about Barack Obama, who was the ex-president of America. His mother was an English, his father was an African. So therefore his body color, Barack Obama's body color is mulatto. He is neither too dark nor he is too white. He is somewhere in the mid. So they are called as mulatto. Okay, so this is for F1 generation. Now how do you obtain offsprings for F2 generation? What you have to do? Now we have to perform selfing, means let a mulatto marries a mulatto. A person who is having 3 dominant allele, let that fellow marry another person who is also having the same combination of gene. Correct? Yes or no? Now how many? Now first of all you have to calculate the gametes. So how many gametes are possible? 8 gametes. Why? Because there are 3 genes. All the genes are present in its heterozygous state. So the value of N will become 3. That is equivalent to 8 gametes. Correct? So beta, how many gametes will be produced over here? Total 8 gametes will be produced. I hope you know the tree method. You can obtain their gametes. Now, once you obtain gametes, then try to make the big Punnett square out of it. And then at the end, the phenotypic ratio, which is going to come over here. Let me just add a page. So, just remember this ratio, beta. You need not to remember the genotypic ratio. Just remember the phenotypic ratio for human skin color is going to be in a range. So, the phenotypic ratio for any polygenic inheritance is going to show a range of phenotype. That is light color, very light color, dark color, very dark color, intermediate. So, there's a range of phenotype. It's not like tall, dwarf, yellow, green, green, yellow. No. Light green, light yellow, more green, less green. So there's a range of phenotype. So here, in case of polygenic inheritance, range of phenotype is obtained. What is obtained, beta? Range of phenotype. Then what is the phenotypic ratio for this? 1:6:20:15:15:6:1. Where here 1 means 6 dominant allele, 5 dominant allele, 4 dominant, 3, 2, 1, 0 dominant. So person with 6 dominant allele will be very dark, will be a Negro. A person with 3 dominant allele will have maximum chances of offspring to have intermediate coloration in that case, which is mulatto. 0 dominant means very light. Now you can imagine very dark, dark, less dark. Then 2 dominant means light, 1 dominant means fairly light, and 0 dominant means very light. So can you see the range of phenotype? Right? We Indians are somewhere here. We Indians either have 2 dominant or 1 dominant allele out of 6. So we are neither too dark nor we are too light. We are somewhere in the intermediate. We call that our body coloration is slightly brownish. Not black, not white. A mixture of that which is slightly brownish. Clear? Yes or no? Now one more thing, beta. The phenotypic ratio of polygenic inheritance can also be shown with the help of a graph, and that graph has to be in bell-shaped manner. So the graph is going to come in bell-shaped manner, where like this you have to show the ratio. 1:6:15:20:15:6:1. So this is kind of a beta bell-shaped curve for the phenotypic ratio of polygenic inheritance. Be it human skin color, many other characters are there which shows polygenic inheritance. Everybody is going to show a range of phenotype which can be shown with the help of graph, and the graph is going to be of bell-shaped curve. Clear? Understood, everyone? This much you have to do for polygenic inheritance. Now, 2-gene interaction is done. We are slightly moving towards from classical, modern. Now we are heading towards the experimental genetics, whose father is T.H. Morgan. Morgan was the scientist beta who actually detected the flaw in Mendel's law of independent assortment. Morgan said that Mendel's law of independent assortment is not absolutely correct because it is having an exception by the name linkage. So study of linkage by Morgan is a part of experimental genetics. We will just have a slight introduction today, but before we start that, there are two more people by the name Sutton and Boveri who tried to copy the work of Mendel by just changing one terminology, that is gene by chromosome. So next theory which you have to study is chromosomal theory of inheritance by Sutton and Boveri. Now beta, these two people did nothing. These two people simply copied the work of Mendel. Whatever Mendel explained for genes, whatever theory Mendel proposed for genes, same theory was used by them, was copy pasted by them, simply editing the word gene by chromosome. So whatever is gene theory by Mendel is same chromosomal theory by Sutton and Boveri. Like for example, let's check out their postulates. Number 1 point, Mendel said that beta genes are always occurring in pair, that they are represented by a pair of contrasting alleles in a diploid organism. So these two people said, of course, chromosome in a diploid organism are also represented in a pair. We know in a diploid organism, for example, there's a human cell. So we human cell are having 46 chromosome. Excuse me. Okay, so human cell is having 46 chromosome, which we say as 23 pair. Why pair? Because every chromosome in a diploid cell has its homologous partner, and on homologous chromosome only the pair of gene is also, the pair of allele called gene is located. So if Mendel said that in a diploid organism a gene is represented by a pair of allele, so Sutton Boveri said that chromosome is also represented in a form of pair in a diploid organism. Correct? So first point, according to them, chromosomes occur in pair, that is a pair of homologous chromosome. Right? In a diploid cell. Second point, Mendel said that beta during the time of segregation, both the alleles of the gene segregate out independently and each gamete is receiving only one allele out of the two. So similarly, Sutton Boveri said that during the time of meiosis, the pair of homologous chromosomes separate out and each gamete receive only one out of the two. Clear? That is why gametes are always pure, they are haploid, because they are only receiving one chromosome out of the pair. So second point, during the time of meiosis, during the time of meiosis, both the chromosome separate out separate and each gamete is going to receive only one out of the two. Right? Third, now Mendel said if you want to make the gene again in the form of pair, then after meiosis, step required is fertilization. Right? Fertilization will only restore the ploidy. Similarly, Sutton Boveri said if you want again that chromosome should come in pair, then next step that is required is fertilization. So third point said to restore the ploidy from haploidy to diploidy. See, meiosis done, gametes are haploid. Now if you again want to go back to diploidy, then event required is to restore the ploidy, comma, fertilization is required. Now, Mendel said that during the time of meiosis and fertilization, alleles of the gene may separate, they may again join back to again form a gene in a diploid cell. During all these events, there is no loss of the gene or gain of the gene, the number remains the same. So same thing was said by Sutton and Boveri that during the time of meiosis and fertilization, the number of chromosome may change. Cell may become haploid, then again diploid, but during all those events, there is no damage to the chromosome. The chromosome number will remain the same. The size, shape and morphology will also remain the same. So you see, Sutton and Boveri did nothing. They actually did nothing. They simply copied the work of Mendel. Wherever Mendel mentioned gene, these two people edited the word gene by chromosome, and now it became a new theory, and we all are supposed to study this theory under the name chromosomal theory of inheritance. Clear to each and every one of you that what is this theory? Great. Now we are heading towards the main experimental genetics whose father is T.H. Morgan, and he actually gave the concept of linkage. So it was studied by Morgan, and Morgan was experimenting on the organism fruit fly, which is scientifically called as Drosophila melanogaster. In today's class, we are not going to start about linkage. I'll be just giving you the hint that why Morgan used fruit fly as one of his experimental organism for study, and the main linkage experiment which this fellow did, we will do in our next class. Clear? So, linkage was studied by Morgan on Drosophila melanogaster. It is basically the exception to Mendel's laws, third law, that is law of independent assortment. Correct? Now, second point which I have written, I will not tell you right now. I'll tell about this second point later when I will teach you the details, the experiment of Morgan on linkage. Today, I am just going to tell you that why Mendel preferred fruit fly. So now, next heading is why fruit fly? Why not another organisms? So, simple reason, the first reason that why Morgan chose fruit fly is that the lifespan of fruit fly is very short. It is nearly 2 weeks. So therefore, the new generations, the scientists do not have to wait long Like if suppose scientists must be working on humans, that for humans the gestation period is about 9 months. So that means scientists will have to wait 9 months for the next generation to come out. So in this case, it is not so. Fruit fly has a very short lifespan, which is of about 2 weeks. First point. Second, the number of chromosomes in fruit fly is very, very less. They only have 4 pair of chromosome, out of which 3 pair are autosome and 1 pair is sex chromosome. Right? Third, they are very easy to culture. Drosophila can grow on any simple synthetic medium. Correct? Fourth, they show same sex determination as humans. That means male Drosophila are represented by XY sex chromosome, whereas female Drosophila are represented by XX chromosome. Now, they show beta sexual dimorphism. That means simply by looking at the fruit fly, we can tell which is male and female. Why? Because males are smaller and females are larger. So first point, why fruit fly? Shorter lifespan, which is of about 2 weeks. Correct? Second point, shorter lifespan, yes. Exhibit sexual dimorphism. That is, males are smaller, male Drosophila are smaller and female are larger. Correct? Next. Third, they can grow on any simple synthetic medium. Scientists need not to worry a lot about their growth. Right? Fourth, the number of chromosomes in a diploid organism is very, very less. 2n is equal to 8. That means 6 are simply autosome and 2 are simply sex chromosome. Next point, they show same sex determination as that of humans. Sex determination same as humans. Like for example, XX is female, XY in case of Drosophila is also male. So these are the certain 5 points. Yes, and one more point, sixth, I can tell you that per mating they produce large number of offsprings. So sample size per generation will be large, hence chances of error is going to reduce. So considering all these such good points, this fellow Morgan chose fruit fly as one of his experimental organism, and yes, of course, he got the success. He actually was able to discover the concept of linkage, and which is also an exception to Mendel's law of independent assortment. Clear? So just leave all these pages. Today we are going to keep till here simple and short. In first 4 lectures, we have done a lot many things as you have already seen. I want you all to revise till there, solve the questions. If you get any problem, trouble, please do not forget to mention in the comment section so that I come to know and I take up your doubts in my next upcoming class. But before I say goodbye, I have some questions for you to solve as homework, which I hope you know the trend. And yes, with this, we come to the end of a simple but easy lecture. Thank you for joining me. See you in the next class. Bye-bye everyone. Take care.